I'm Rishan.
I'm Chenjie.
I'm Singi.
And I'm Emily.
Hi guys. So, welcome back. Today we're going to talk about a topic that actually is a very serious issue in the current world right now, which is the challenges being faced in modern agriculture. So, right now, farmers across the globe are dealing with like two major threats, man. So one is like being climate change, and the one being rising pressure from pests and plant diseases. And honestly, it's hitting food security pretty hard right now. And we're seeing higher temperature, unpredictable rainfall, and more extreme weather all across the globe, which is affecting a lot of crop yields. Take wheat and maize, for example. The heat stress alone can mess with the pollen viability and grain development. That actually leads to much lower harvest, so less food for us. And at the same time, pests and disease are getting smarter and stronger. Warmer climates let the disease spread to new regions, and some are now even resistant to chemical treatments. And also there's a study by Savory in 2019. It actually showed that crops are losing their yield, especially in bad areas where new or reoccurring pests are common. So the bottom line of this is basically our current methods, they aren't cutting it. So if we want to protect our food system, especially in these like important vulnerable areas, we're going to need something fresh. We need new ideas and a sustainable solution that is able to help crops adapt to both environmental stress and biological threats. So what do you think about that, CJ?
So with all the pressure on agriculture from climate change, pests and plant diseases, relying on traditional methods alone isn't enough anymore. So that's why tools like functional genomics and proteomics are gaining so much attention in the crop science now. So to keep it simple, functional genomics help us to understand which genes are active and what they actually do, especially those linked to traits like heat tolerance, disease resistance, or better yields. So while proteomics, on the other hand, they help us to look at the proteins that those genes produce, which are the actual molecules that work inside the plant for handling stress response in real time. So you can think like, if the gene is the blueprint, the protein is the worker following the instructions. While genomics tell us what could happen, proteomics tell us what is happening at the moment. So that makes them powerful when used side by side. So by combining these approaches, scientists can dig deep into how plants behave under stress, like drought, high temperatures, pests, or disease, and figure out which genes and proteins help them survive. And once we know that, we can use those genes and proteins to guide crop breeding or even genetic engineering. So it's all about making crops that are not just productive but also resilient in changing environments. So now that we have looked at what functional genomics and proteomics are all about, let's take a closer look at how genomics is actually applied in real crop research.
Let's take a closer look at how functional genomics is changing the game in crop improvement, helping scientists build better, stronger, and more nutritious crops from the inside out. So with the help of tools like gene sequencing and gene activity study, scientists today can find the exact genes that control important growth traits like how much a plant produces and how well it handles stress and how healthy the food is.
Interesting. Can you give an example like a real crop?
Sure. Let's take rice. One important gene linked to better yields is GN1A. These genes make an enzyme that breaks down a plant's hormones called cytokinins, which help plant grow. So when the activity of these genes is reduced, more cytokinins stay in the plants, helping it grow more branches and produce more grains without making them smaller. Another key gene is GS3, which controls grain length. It normally keeps grains short by slowing down cell growth, but when scientists lower the activity of these genes, the grains grow longer, which is something many farmers prefer.
So how actually do they test or change these genes?
Scientists use tools like CRISPR, the genes knockout methods, and even mutant plant study to test these genes. So once they know which one works best, they will use marker-assisted selections to breed better plants more efficiently. So in one case, editing the GN1A genes increased grains number by up to 30% in certain rice types. But improving yields is only part of the story. So genomics also help us understand how plants deal with difficult conditions like drought and salty soil. So for example, in rice, scientists found genes that affect how the plants respond to stress, like OSD1S1. If this gene is too active, the plant becomes more sensitive to drought. But when it's turned off or reduced, the plant survives better with something show over 50% more survival in dry conditions. So for example, in tomato, a gene called SIMPR1 was found to help with drought resistance too. So when scientists turned these genes off, the plant lost more water and were more easily damaged, showing how important the genes is for surviving tough weather.
So what about nutrition? Can genomics help make crops healthier?
Yes, genomics also help make food healthier and safer. So in potatoes, researchers added a gene called VINV to stop sugar from building up during cold storage. This lead to 90% less sugar and over 70% less alkali amide, a chemical that can be harmful when fried. So and finally, there's a golden rice, a type of rice designed to make vitamin A. Scientists added 2 genes, one from corn and one from bacteria, so the rice can produce beta-carotenes. So this is very helpful in areas where people don't get enough vitamin A from their diets. So overall, genomics help us understand and improve crops from the inside, whether it's making food, helping plants survive tough conditions, or making food more nutritious. This science is helping build a better and more secure future for farming.
Thanks for that great overview on how genomics is helping us grow better and stronger crops now. Let me take you a little deeper, not into how we grow crops, but how we protect them. You see, plants don't just grow on peaceful fields under blue sky, they were constantly under attack from bacteria, viruses, fungi, tiny worms, and insects. These attackers can cause small infections or they can wipe out the entire harvest. One example is the dieback disease in Java trees in Western Australia. It is caused by a fungus in the soil and it has led to large-scale destruction of native forests.
So how do we protect plants from things like this?
Yeah, so this is where genomics once again plays a critical role. Just as it helps us improve growth, it also helps us to understand how plants defend themselves. Think of it like decoding a plant's immune system. By studying a plant's genetic makeup, scientists have been able to identify specific genes that helps plant recognize and respond to invading pathogens. These are sometimes called resistant genes. Think of them like an early warning system. When the pathogen shows up, the plant activates its defenses. We have learned that for every resistant gene in a plant, there is usually a corresponding attack gene in the pathogen. This concept, known as the gene-for-gene relationship, has helped researchers to develop new ways to breed or engineer disease-resistant crops. In canola, for instance, researchers have identified 23 resistant genes that help the plant fight off fungal diseases like blackleg disease. With the help of tools like microarrays analysis and next-generation sequencing, scientists have mapped how plants like peas and legumes respond during infection. What signals are sent, which hormones are activated, and what defense chemicals are produced. It turns out that plant resistance isn't just one switch. It's a complex, layered system involving structural changes, chemical responses, and coordinated gene activity. However, beyond understanding, we can now act. One of the most exciting tools in this area is CRISPR, a precise gene editing technology. It allowed scientists to make targeted changes in a plant's DNA and, for example, like enhancing resistant traits or removing genes that make the plant more vulnerable to disease. This has already been shown to protect crops like tomatoes, soybeans, and peas from various fungal and bacterial infections. Besides, CRISPR can be used to weaken the invaders themselves, for example, editing a gene in the pest Helicobacter armigera, a common caterpillar that damages crops, can make it more sensitive to natural toxins or reduce its ability to absorb nutrients. This kind of targeted pest control reduces the need for chemical pesticides. In short, genomics isn't just helping us grow better crops, it also serves as a defense system, equipping plants to survive the threats they face every season.
So why does this matter for the environment and the economy?
Well, when crops are naturally resistant to disease and pests, farmers can reduce their use of pesticides and fungicides. That means less chemical runoff into rivers and soils, fewer risks to pollinators like bees, and safer food. Economically, it means fewer crops losses, more stable harvests, and lower costs for farmers, especially in developing regions where resources are limited. Furthermore, it also means resilience. Genomic tools help us breed crops that can handle not just disease, but also drought, poor soil, and changing climates. That is essential as we face more unpredictable weather patterns and growing food demands. It's a smart and sustainable way to tackle an OH problem, and because these edits are so targeted, they don't mess up the rest of the plant. That means we can grow crops that are naturally strong against disease and pests with less chemical use, less environmental damage, and more reliable food production.
Thanks for the great insight on the environmental and economic benefits. Now let's shift to the social side of things. Genomics-driven agriculture holds powerful potential to improve life, starting with food security. So by creating high-yield and climate-resilient crops, we are helping communities maintain stable food supply even in the face of climate change. And it doesn't stop there. So through biofortifications, we can enhance the nutritional values of everyday staples like rice and wheat, so helping fight malnutrition in areas where it's needed most. So on the top of that, empowering farmers with access to these improved crops support local livelihood. So it gives communities the tools to become more self-sufficient, healthier, and more resilient for generations to come.
Alright, thank you Singi and Emily. So we have seen what the genes can tell us. So now it's time to see what the proteins are doing behind the scenes. So it's amazing how much information you can get by just looking at protein changes in plants under stress. So for example, in a study on drought stress rice that I found, researchers used proteomics and found proteins like DHAR1 and LTP1 were upregulated, meaning that their levels increased during drought stress. So don't worry too much about the name. What matters is that these proteins help to detox harmful molecules and protect the plant cells. So because of this, they are now being looked at biomarkers to breed more drought-tolerant rice in the future. So just by knowing what proteins show up during stress, we already have a target to work with.
So that's really cool. Are there any examples for other plants?
Yeah, so there's another interesting case in soybean where researchers compared a stress-tolerant variety with a sensitive one under heat and water stress. So they found out that the tolerant soybean had higher levels of antioxidant enzymes like superoxide dismutase and peroxidase. So these proteins, they act like the plant's built-in cleanup crew. So they help manage oxidative stress, which builds up when the plants are under stress. So it's kind of like it is the plant's own fire department. They putting out the damage before it spreads. And then there's weight. So researchers look at how the plant responded at low oxygen conditions, which can happen during waterlogging. So they found that the tolerant wet plants had more proteins like alcohol dehydrogenase and SAM1, which help the plant maintain its metabolism when oxygen is limited. So that kind of precision at the molecular level, seeing exactly which proteins get turned on in tough conditions, really shows the strength of proteomics. So what's really cool is that proteomics don't just say, okay, this plant is stressed. So it actually tells us how the plant is trying to deal with it, like which pathways are activated or which defense system kick in. So, and I think most of us came across something like that in our research, where it's not just about reviewing the stress, but about understanding the plant's actual survival strategy, right? Yeah, so and another thing is that stood out to me is how this study often lead to the discovery of biomarkers. So these biomarkers are proteins that consistently show up in stress-tolerant varieties, and they can be used to screen for good traits in breeding programs. So instead of growing thousands of plants in the field just to see which one perform best, researchers can use these biomarkers to identify which plants have the traits they are looking for. And all of these examples show how proteomics lets us zoom in and see which proteins switch on when the plant is under stress. And from there, we can start designing crops that handle those pressures better. And that's what makes proteomics so valuable. It helps us discover which proteins are really doing the heavy lifting during stress, and give us real functional information that can be applied directly, and whether it's in macro-assisted selection, precision breeding, or even gene editing, and it's more accurate than traditional methods and can save years in the crop process. So it's exciting to think how this kind of research could help farmers in the long run, like growing rice or soybeans that can handle drought or pests better without needing as many chemicals or inputs. And that's what actually sustainable agriculture is really about, right?
Yeah.
I
So
think
it's
that's
better for the environment, better for food security, and also better for the people who depend on these crops.
You're absolutely right, CJ. Proteomics is such a powerful omics technology, you know. It's transforming the way we're able to understand and improve the nutritional quality and safety of our food. Of course, I mean, we all know that proteomics is all about studying the full set of proteins, in this case, plants. So, what are proteins? Well, it's not only because they're essential in nutrition, but it also can affect how food tastes, how it feels, and if it's safe to eat or not. So, when it comes to nutrition, actually, proteomics helps researchers identify what proteins are actually linked to these valuable nutrients. You know, think essential amino acids, vitamins, antioxidants, you know, the good stuff for your body. But by comparing different type of crop varieties, scientists can, you know, further zero in on the proteins responsible for producing these compounds.
I see, that's actually quite interesting.
Yeah.
But has it actually been done before?
So, an example, huh? So, from the top of my head, I can think that, well, some rice varieties actually have been studied for their ability to naturally accumulate more iron and zinc, which is quite important with the global malnutrition problem. But you have to remember, it's not just about the nutrients that are present. It's also about how we're able to absorb those nutrients. Is it good or not? So like some proteins like lectins or phytic acid, they actually can block the nutrient uptake. And proteomics actually can help detect these anti-nutritional factors and guide us in finding ways to minimize them, whether that be through breeding or better processing techniques. In fact, processing itself is a huge area where proteomics is making the difference. Studies have shown that cooking and preparing food can change protein structures, you know, sometimes helping digestibility, sometimes not. But protein analysis also help us see what exactly is happening at that molecular level. So we can actually make some tweaking in the method so that we can get the most nutrition out of every bite.
Oh really? But nutrition is only the story, right? So food safety is another major factor?
Absolutely right. Yes. In food, you know, we have many allergens, you know, like milk, nuts, eggs, you know, mainly proteins. And even trace amounts of these proteins can actually trigger serious reaction. Proteomics, especially mass spectrometry-based tools, can actually detect these allergens with, you know, impressive sensitivity. And even in processing foods where the proteins are broken down, that means better labeling, safer product management, and of course, peace of mind for the consumers with allergies.
Wait a minute, but I have one question. What about being able to detect harmful microbes and toxins?
Well, actually a very interesting case right here. Proteomics can do that too. You know, like an example is actually if a food is contaminated with like bacteria such as Listeria or E. coli, very harmful to us. Their unique signatures can be identified early, sometimes even before the bacteria cause any symptoms of spoilage. So at the same time, it goes for fungal contamination. By spotting fungal proteins, we actually can act before these dangerous mycotoxins build up.
Oh, I see.
Yeah. So what makes proteomics especially valuable in this sense is actually its speed, accuracy, and its ability to work in complex food samples, whether that be pinpointing allergens, tracking toxins, or mapping out nutritional qualities. It's a behind-the-scenes hero type of thing that helps make our food healthier, safer, and...
So like, knowing all this, what does it mean for the future of modern agriculture?
As we look to the future of agriculture, genomics and proteomics are set to become even more powerful, thanks to new technologies like artificial intelligence and multi-omics integration. AI and machine learning are now helping scientists to analyze complex genetic data faster and more accurately. This means we can identify useful traits like drought tolerance or disease resistance more efficiently, speeding up the crop breeding process. At the same time, multi-omics approaches are giving us a deeper, more predictive view of plant biology. By combining genomics with proteomics, metabolomics, and phenomics, we can now predict how combinations of genes, proteins, and metabolites interact to shape complex traits like yield, flavor, or stress resilience, making breeding more targeted and efficient. Together, these innovations are paving the way for smarter, more sustainable living, more sustainable farming, where crops are tailored for performance, resilience, and reduced environmental impact.
In the long run, genomics and proteomics-driven agriculture have a huge potential to help the world reach important global goals like ending hunger, improving nutrition, and protecting the environment. By creating crops that grow better, survive tough climates, and carry more nutrients, we can support both farmers and communities around the world. These solutions are smart, long-lasting, and can be adapted to different regions and needs. So in the end, genomics and proteomics are not just about the better crops. It's about building a better, fairer, and more resilient future for our planet and the people who depend on it.
So that's the end of the podcast. Remember, tell your mom, tell your dad, tell your friends, tell your pet dog. Please like, subscribe, share to everyone you know. Alright, so that's all from us. See you all and
Bye!